Hi everyone.
Nice to see you.
Well, the problem with metamsodium is if you use it consecutively for a number of years, you start to get problems with microbial degradation. And how that works is as the metamsodium breaks down to its active ingredient, which is methyl isothiocyanate, you get an increase in microbes that quickly break down your active ingredient before it starts affecting the pathogen. So it basically renders the metamsodium inefficient.
Another problem with soil sterilants is that you get very inconsistent results. Various studies have shown very different kind of results. Some studies do not find any differences in the bacterial diversity, but that does affect the microbial richness and fungal diversity maybe. And sometimes you get higher potato yields, but only in sandy soils, whereas you get the opposite trend in loamy soils. And then other times, treatments did not result in an overall shift in microbial community structures. But what is very important is Fusarium is one of the pathogens that is being controlled with soil sterilization. And one of the studies did not find any effect on Fusarium itself, which makes it kind of not very efficient to use this method at all. Another problem with soil sterilization is the huge input costs in this method of pathogen control, and the fact that some of these sterilants will be phasing out of the market as well.
Okay, so to understand how long soft rot bacteria survive in the soil, we need to understand how it lives in the soil. So it typically exists in asymptomatic form as latent infection of potato tubers, or as symptomatic, that's when we start to see symptoms. Now, if there are plants in the soil, whether it be symptomatic, non-symptomatic, weeds, ornamental plants, it can last longer because there is a host that is hosting it. So that's the first problem. So having plants in the soil influences how long it will survive. The second point is that the host, so soft rot bacteria are typically broad host spectrum, so they infect literally anything. So there may not be potato tubers in the soil or potato plants in the soil, but whatever plant that they typically can infect will allow them to survive longer. Now the last scenario is looking at plain soil. If there is only soil, no plants, then they will survive a little bit shorter. This can be from 1 year to 3 years. In our lab, we work a lot on Pectobacterium, and we see that it can be about a year. But reports from Europe, where they find mostly Dickia, find that it can be a little bit over a year.
So the main causal organism of Verticillium wilt is Verticillium dahliae. And as you can see, here's a picture of Verticillium dahliae. This is the microsclerotia, which can typically survive in the soil between 10 and 15 years. And this is in fallow soil. That's now without the absence of a host. And then Fusarium wilt is caused by various Fusarium species. And Fusarium forms chlamydospores, which can also typically live between 5 and 10 years in the soil with the absence of the host. So both of these organisms can survive for quite a long time. So you will need very long rotation periods between your potato crops. Verticillium wilt was mainly caused by Verticillium dahliae. And as you can see, here's a photo of Verticillium dahliae microsclerotia. And this structure typically survives in the soil between 10 and 15 years in the absence of the host. And then Fusarium wilt is caused by various Fusarium species. And the Fusarium forms chlamydospores, which typically lives in the soil of between 5 and 10 years in the absence of the host. So as you can see, both of these fungi can survive for quite a long time. So you need to have long rotations in between your potato crops.
If we look at Streptomyces scabi, or the causal agent of common scab, we can see that this bacteria can live as a saprophyte in the soil. Therefore, with this, it can live or survive in the soil for extended periods. What makes it even worse is that the pathogen can survive on stored seeds in seed warehouses. If we look at bacterial wilt that's caused by Ralstonia solanacearum, it has an alternate host that is very large. It can also survive in irrigation water and soil, but in soil it goes into a viable but not culturable stage, which is difficult to detect. It is believed that this pathogen in a fallow land can survive between 8 to 10 years.
So Rhizoctonia solani is a fungal pathogen that can also survive in the soil for a long time. It has sclerotia that are the survival structures of this fungus, and it can survive in the soil for up to several years, depending on the environmental conditions. Another serious fungal pathogen of potato is Scolototricum cacoides. This pathogen also forms sclerotia that can survive up to 10 years or even more in the soil. So this pathogen can actually cause stem cankers and wilting, as well as a post-harvest blemish on the potato tubers called black dot.
So these are the results of the study that I did, where I did several green manure applications to look at the effect of the green manure crops on the development of verticillium wilt on potatoes. And I measured it by looking at the development of verticillium wilt on the potatoes. So you can see I used a disease severity rating of between 1 and 5, where 1 was healthy plants and 5 was a dead plant.
So when we started sampling at the Eastern Free State trial, we were looking to see whether there is Pectobacterium in the soil as our first objective. And we also looked at the diversity generally of bacteria in the soil. So what you see on the slide are the typical isolates that we got. And you will see that mostly they represent plant growth promoting bacteria, such as Bacillus, Pseudomonas, and Pantua.
Well, with the addition of compost and biological control agents, we actually didn't see an increase or decrease in the disease severity, but actually there was an increase in the enzyme activity. Usually with the increase in enzyme activity, there's an increase in soil diversity and more competition for the pathogen. So on the long term, adding compost or biological control agent will actually decrease the disease. In another study that was done at the ARC by Dr. Renette Gous, she actually see that the mustard as well as brassica species actually inhibit Streptomyces spore. She also built a potato split root unit where there was a decrease in the Streptomyces population. She also saw that brassicas shoot tissue inhibit the growth of Streptomyces species, and that several other brassica species also inhibited the pathogen.
So you'll remember that I said earlier that we isolated bacteria that have plant growth promoting attributes, such as Bacillus. So they use, to increase the plant health, they use mainly 2 main mechanisms. The first one is that of antagonism, or killing or inhibition of pathogenic bacteria. So on this slide, I show you a picture of Pectobacterium lon, on top of which we plated a Bacillus species right there in the middle. What you see there, the clearing zone, indicates that the Bacillus are producing enzymes that have ability to kill Pectobacteria. What is really nice is that this inhibition is not limited to just bacteria, but it also extends to other pathogens such as fungal and oomycete pathogens. So that's the first mechanism, that of inhibition. The second mechanism is where the bacteria boosts the plant's immunity. For example, through induced systemic resistance. One could think of this as a non-pathogenic bacteria colonizing or infecting a plant. It actually doesn't cause disease, but because it's a bacterium, it has similar features as those that are pathogenic. So the plant gets early recognition of a microorganism on it, and that starts to boost up the resistance, so that when pathogens infect, then the plant is more resistant. Other mechanisms include things like siderophores. So these bacteria are very good at scavenging for nutrients that the plant needs but is not able to get, such as iron. The other well-known mechanism is nitrogen fixation, so the bacteria can help the plant fix nitrogen when the plant doesn't have that ability. And lastly, it also allows for the plant to produce hormones that are involved in resistance.
So as I mentioned previously, I looked into the application of green manures for the management of verticillium wilt on potatoes. So typically green manures, the mechanisms involved with green manures is the shift in the microbial population that happens after the application of the green manures. The second mechanism is the plants that are healthier due to the application of the green manures. And then also the release of volatiles. So I just want to go into the volatiles a little bit. This is a study that I did during my PhD a couple of years ago, where I looked at several different green manure crops, and some of these are also used in my study for Potato South Africa, and that is black oats, sorghum, Sudan grass hybrid, and white buffalo grass and white mustard. During the study you can see that the no amendment control had 100% germination of Verticillium dahliae microsclerotia, and that black oats, dolichos beans, the sorghum Sudan grass hybrid, the white buffalo grass and the white mustard all inhibited the germination of the Verticillium dahliae microsclerotia to such an extent that it only below 10% of the microsclerotia germinated.
So another mechanism of inhibition of pathogens is basically by increasing the soil microbial activity. Now, one way to do this is planting a mixed cover crop. So the different plants will attract different microorganisms. So the higher diversity of microorganisms in the soil will lead to higher competition with the pathogens. And so hereby you will also be able to reduce the disease. So another way of inhibiting the plant pathogens is increasing your soil microbial activity. And one way of doing this is planting a mixed cover crop. So the different plants will actually attract different microorganisms, and this increased diversity in the soil will lead to higher degree of competition with your pathogens, in turn which will lead to lower disease levels.
So for future control strategies, I would definitely say that we should focus to increase soil health, and we should focus on the natural disease suppressiveness of the soil. You have to carefully select your crop mixtures for cover crops and green manures. Some of the cover crops and green manures have good characteristics such as legumes that can enhance your nitrogen in the soil. And also each crop has its unique microbial diversity living in its rhizosphere, which can lead to an increase in natural enemies in the soil.
I definitely agree with Estienne. We need to improve soil health. There isn't a golden bullet that will solve all the problems. What perhaps she missed that we can add is biological control agents, your PGPRs, your mycorrhiza, that we can also add to increase our soil health.
Clearly, these cropping systems are bearing fruit and they are increasing plant health because in our case we didn't find any Pectobacterium. They're also enriching the soil of beneficial microbes. And there are also long-term benefits such as carbon cycling, water cycling, nutrient cycling. So in the long term, I see a possibility of using all of these biologicals, perhaps in combinations with chemicals for a short while they're still being developed.
I definitely agree with my colleagues. We need to move to a more biological way of controlling diseases and increasing the soil health. Part of the reason for this is that we are losing more and more chemicals, and more of the pathogens are developing a loss of sensitivity to the chemicals that we are still using. So the biological control will have to be increased, and to do this, especially in the soil, we have to increase the organic material in our soils. A lot of the soils is very dead, which means it cannot even sustain good organisms that you're adding to the soil. For this to work, you have to increase the organic material in the soil, and cover crops, green manure and biological control is definitely the way to go.